much for the kind introduction and also for the opportunity to be here today and present to you my work, my research work, which is currently focused on detecting changes in brain connectivity across many different neurodegenerative disorders. I will speak mainly of Alzheimer's disease. And to detect these changes, we typically use brain imaging techniques and also other kinds of biomarkers. So as was just mentioned, and for those of you who not know me, I'm an associate professor at Karolinska Institute here in Stockholm, specifically at the Neuro Division of the Clinical Neurosciences Department, where I currently lead a group of 3 PhD students, 3 postdocs, 1 research assistant, and 3 clinical associates. And I also supervise 4 PhD students, 1 from KTH and the remaining ones from Gothenburg University. So most of our recent research can be summarized into 3 broad categories that actually correspond to 3 ways of analyzing brain connectivity. So the first one, we usually refer to it as spatial patterns, which are groups of brain regions that strongly correlate with each other, forming what we call a covariance network. The second one was already briefly mentioned by Peter, as I suspected, and is network topology, which we use to assess different properties of brain organization using a mathematical framework known as graph theory. And finally, we also look at synaptic markers, which can be extracted from the cerebrospinal fluid of the patients by a certain needle between the vertebrae in the back. And these are quite useful to assess at the whole brain level the levels of synaptic proteins, as well as other proteins that might be relevant for our disease. And of course, the synapses are the smallest brain connections you can imagine. So when it comes to brain imaging, I would just like to highlight one point. Most of our research is focused on MRI and PET imaging techniques. And I was sort of warned by the organizers of this event that perhaps most of you are interested in MEG or EEG. And I would just like to say that I've not yet had the pleasure, or I'm starting slowly to getting to know a bit more about these imaging modalities. But all the techniques that we'll be presenting today, that can be perfectly applied to MEG or EEG data. It just takes a little bit of imagination. When you see MRI, you think of MEG. When you see a PET, you think of EEG. Yeah, the results will be different, but yeah, it will be fine. Okay. So when it comes to spatial patterns, these have been analyzed using different imaging modalities, but we focus mainly on an imaging modality in which maybe these spatial patterns are not so well known. This imaging modality is called positron emission tomography, or PET imaging, and it consists of introducing a radioactive tracer through the bloodstream of the patients, which circulates and crosses the blood-brain barrier and binds to proteins in your brain that show an abnormal configuration that have somehow, for some reason, become pathological. Okay, for those of you who are not so familiar with Alzheimer's disease, the only thing that I really need you to know is that it is, of course, a neurodegenerative disorder. I didn't feel that one. But again, I'm more used to saying the word than you are. And it is associated with the abnormal accumulation of 2 kinds of proteins. So if this was working, I could point you to the proteins. One of them is amyloid beta, and the other one is called tau. And these are proteins that are normally present in our brain, but for some reason that we haven't managed to figure out yet, they become abnormal in Alzheimer's disease. They start playing their normal functions and they start deteriorating the way the brain is working in these patients. So these 2 proteins seem to spread in the brain according to a very characteristic spatial pattern as the patient progresses through different stages of the disease, with amyloid starting first in neurocortical regions, from which it extends to limbic areas and finally reaches subcortical regions. Whereas in the case of tau, is not exactly the same spread of pathology. So it typically starts in the locus coeruleus and entorhinal cortex, from which it extends to other limbic regions and finally reaches the neurocortical areas. So thanks to the relatively recent development of amyloid and tau PET tracers, it is now possible to assess their spatial patterns in vivo, which provides us a unique opportunity to see how these patterns look like and also how they are related to one another across different disease stages. And because of this, in this first study, we were very interested in measuring the spatial patterns of tau pathology. And for that, we used a tracer that is called fluorotaucipir. And it's been shown to be a very good tracer to reflect underlying pathology because it actually correlates with the levels of intrasomal tau and also of tau-positive neurites. So this is just like a proof of concept that the tracer that you are using is actually measuring the pathology of the disease. So there is increasing evidence suggesting that when you measure the tau signals from a tau PET image, for example, in two different brain regions, these signals are not independent from each other. In fact, they are strongly correlated, forming what we call a spatial pattern or a covariance network that could look, for example, like this one. And one very common method to identify a spatial pattern is by using independent component analysis, which is are best known in the field of fMRI and perhaps MEG and EEG, but they can also be applied to static data that does not show variation over time. And it's very good to identify subcomponents within the data that potentially represent meaningful networks that you can use for posterior analysis. So motivated by this evidence, in this study, we set out ourselves to identify the spatial patterns of tau pathology in a collaboration with Berkeley University. And we did this in a sample of cognitively normal older adults, some of which had a higher risk to develop Alzheimer's disease. And much to our surprise, because tau pathology is usually observed in more advanced disease stages, much to our surprise, using an ICA, a simple ICA analysis, we could already observe 10 clearly different tau patterns. So above you can see there were, oh, it's not showing the entire slide. I noticed it now, but the figure above represents, I hope it doesn't, it's not a problem for other things, but I'll keep going. Yeah. We noticed there were a couple of temporal patterns. Their names you cannot see right now, but I promise you they're there. But let's go with that. Let's go with trust. I think it's the best thing we have right now. And there were also a couple of bilateral occipital patterns, a couple of frontal, and even a sensorimotor pattern, which was a little bit surprising because sensorimotor areas only show tau pathology in very late disease stages. So once we identify these patterns, what we did was to extract the tau signals from each of these components and try to see if they were related to anything, if they were meaningful in any way. And we observed that indeed they were associated with retrospective cognitive decline over a period of 10 years, in particular in executive and in memory functions, which are of course important for Alzheimer's disease. So we thought this would be a good way of assessing tau pathology and their maybe its association with cognitive functions in the context of Alzheimer's. But it could be applied to any other disorder, of course, and with other PET tracers or other kinds of tracers. Okay, another thing that we did with the SIM study was to, let's say, rank the spatial patterns that I showed you before according to the frequency of tau positivity. So we established a threshold above which we decided that this scan is tau positive. So this individual has a higher risk of developing the disease because we were just interested in seeing which patterns are more common and followed by which ones, and does this progression overlap in any way with what has been seen in postmortem studies. So the most common pattern included medial temporal regions, which are involved early in the course of Alzheimer's disease, which were followed by ventral occipital and orbitofrontal regions. Also, a few additional medial temporal regions that were not included in the first pattern. Lateral brain areas from the temporal and parietal cortex, the medial parietal cortex, the superior frontal gyrus, the sensorimotor cortex, and finally the frontal pole. So when we compare the progression of tau pathology across these spatial patterns, we found them to agree reasonably well with the spread of tau pathology previously reported in postmortem cases. So we thought that this would be a good way to get an insight into the pathology in vivo of these individuals, particularly at these early disease stages. We thought that was quite nice. In a subsequent study, we were still interested in the networks or components derived from tau PET imaging using the same tracer, but here we added an additional aim, which was to identify also the patterns associated with the other pathology of Alzheimer's disease, which is amyloid. And for that, we use the fluorobeta tracer, which is also shown to be very well associated with amyloid plaque burden in postmortem studies. So again, agreeing that this is a good marker for underlying amyloid pathology. But here our aim was slightly different. So instead of identifying components in the separate imaging modalities, we're interested in seeing in the components there were identified jointly that were linked between each other, since these two pathologies are supposed to be associated during the course of Alzheimer's disease. And for that, we identified the joint patterns using particularly a joint ICA analysis. So it's a normal ICA analysis, but it sort of determines the hidden structure in the data by using the matrices from the two imaging modalities. And here using the joint ICA, which was calculated using the fusion toolbox of Vincent Calhoun, we identified links in the two imaging modalities between different areas and tried to find these joint patterns for the two pathologies. So one of our first results was that many of these patterns for amyloid on the left and tau on the right, so a few of them, not many, a few, overlapped in similar brain regions. So for example, we had an inferior occipital pattern, a superior frontal one, and a medial parietal one, which we thought was interesting. But what was really surprising was that most of them did not overlap in the same brain regions. Despite being linked or strongly associated with each other, they were not overlapping in similar brain areas. Again, it's quite unfortunately, you cannot see the superior part of the slide, but these were the components that were linked for tau and amyloid across different components and disease stages of Alzheimer's. So from these results, we could conclude that amyloid and tau pathologies are not targeting the same spatial networks in the brain. Within the same study, we also had access to some resting state functional MRI data. And therefore, we thought we could also see how these pathological patterns overlap with these networks. And to our surprise, we observed that the amyloid patterns, which are located over here, showed a preferential overlap with the anterior and posterior components of the default network, which, as you know, is a very important network for cognition that is found to be often altered in many neurological disorders. Where when it comes to tau, it seemed to be less selective. So the tau patterns seem to overlap with a wider range of resting state networks that included the limbic network, somatosensory, or, for example, the right frontoparietal network. So it seemed that tau overlapped with networks that were a little bit more distributed. Within the same study, we were lucky to have some diffusion-weighted data, which is important to extract the integrity, for example, of the white matter tracts that connect the areas of the brain. And here we observed that in contrast to the previous results, only the tau patterns were associated with the loss of integrity in these anatomical connections across the different groups of subjects at risk for Alzheimer's disease. So this was quite an interesting finding. And to conclude, this study showed that amyloid and tau pathologies seem to target distant networks of the brain, and therefore it is not surprising that they also overlap with different functional brain networks. What we did find surprising was that tau was the only pathology that was associated with reduced integrity of white matter tracts, but this is actually in line with this biomarker being more closely related to neurodegeneration and cognitive function in Alzheimer's disease. So you know this disorder is characterized by different changes, and tau is sort of a later marker. So it's closer to the diagnosis of dementia compared to amyloid, which could potentially explain these results. Now, changing a little bit the subject to something that Peter has already mentioned before, is network topology. And this is something I've always been very interested in because I think it can be used to understand many things in our environment and not only our brain. So to illustrate this notion, I've included here two buildings that, despite having a different color, they look exactly identical from the outside. So if we take a look at the architecture or the inside of these buildings, we can see that despite the fact that they are formed by the same number of pillars or these sort of walls, I actually drew them myself, I don't know how to call them, we can see that these pillars are organized very differently in one building compared to another, which in most cases would not make any difference. Both buildings would be perfectly functional, people working there every day, having fika, joining with friends and so on. But in the unfortunate event, for example, of an earthquake, it can mean that one of these buildings becomes pretty much destroyed, whereas the other remains intact. So the reason why I included this is because the same thing happens with our brains. Two brains can have the same number of connections, but the way they are organized can make one brain much more vulnerable to develop a neurological disease. This is something important to keep in mind. To further illustrate this, but now specifically apply to the brain, I've included here three types of networks, which, as in the example before, are all formed by the same number of connections, and they all have the same number of regions, which, as Peter mentioned, we refer to as nodes and edges in graph theory terminology. So as in the example before, you can clearly see that these networks have connections that are organized in a very different way, with the one on the left having a lot of connections between regions that are close to each other, which even form these triangles between them. Whereas the one on the right is sort of the opposite network. It's a network in which most of the neighboring areas are not well connected with each other, but instead areas that are far away from each other are connected by direct links, direct connections. So we know this now from many years of research and hard work, that in order for our brain to function properly, we need an organization that lies between the two. So something that looks more like this one. So we need our neighboring regions to be well connected with each other, but we also need a few long paths that work like shortcuts and allow you to jump from one distant point of the network to the other without having to go all around it. So this is what a healthy brain network works like, which I hope that is the case of most of the people present in this room. So this balance between what we call local connectivity between neighbor regions and global connectivity between regions that are far away from each other is very important and can be assessed using different graph theory measures. Here I'm going to highlight only 3 of them. One of them was already mentioned, which are the paths. And the paths are simply the distance between 2 brain regions in terms of connections. So here we have region A and region B, and we can see that they are connected by 1, 2, 3, 4 paths. So they're fairly distantly connected, these 2 regions. Another important measure is the clusters, which are reflecting how well a specific node is connected to its neighbors, and also the neighbors are connecting to each other, forming these sort of triangles between them. And finally, another measure that was also mentioned is the models or communities, which are groups of brain regions that are very tightly connected within themselves, but not so much with the rest of the network, which was something that puzzled people initially. But now we know that this corresponds to the ability of the network to specialize in a specific function. So when we measure these models, we found that they often correlate with a specific cognitive or behavioral function. So it's a way for the network to specialize itself in doing something really well. And by the way, your network can specialize in doing things all the time. So if you happen to repeat a certain activity over a few weeks, you will find that, if you scan those connections, you'll find them to be reorganized, which is something I find truly fascinating, particularly for a person like me who's already old. So this is something that happens. But what can these measures tell us about a complex disorder such as Alzheimer's disease? They all have their meaning. They seem to be important, but what can they really tell us that is clinically meaningful? So one thing that we found was that when it comes, for example, to the paths or the distance between brain areas, these seem to be really useful to detecting the earliest stages of the disease. And by earliest, I mean individuals who are completely cognitively normal. They show no signs of Alzheimer's disease, but we know from their biofluid analysis that some proteins are already abnormal, particularly they have signs of amyloid pathology. And by the way, nowadays you can diagnose this, you can detect this up to 25 years before you're diagnosed with a disorder, which is both scary and, scary, I would say. So here in this study, we had access to a very large cohort of cognitively normal participants. And these individuals, all of them underwent diffusion-weighted imaging. So first, we represented their brain areas using an atlas. And here you can see the nodes, which is something that we name brain regions in graph theory. And then we identify the links between these nodes using the white matter fibers between them. So at the end, what we got was a brain graph that looked pretty much like this one for every single individual. And one of the things that we observed is that when we measured the distance between the brain regions, coffee machine is working, that's good. When we measured the distance between brain regions, we found them to be very much increased in those individuals that had a higher risk to develop Alzheimer's disease. Which means that the regions of the network were probably not communicating so well with each other. And very interestingly, these increases in the distance between brain regions correlated with worse memory functions, which happens to be one of the first clinical symptoms of Alzheimer's. So we thought that these findings were quite encouraging, when it comes to detecting very early changes in this disease. Another thing that I've included here, which is a little bit different, it also concerns the distance between brain regions and some results we obtained with this measure. But here, what we did with graph theory was something slightly different. So instead of building a white matter, a gray matter network, we built a pathological network, which is something that you can also do. So a pathological network is a network in which regions, the information that they contain is perhaps, for example, the protein levels that are abnormal in a certain group of patients. So it's also possible to build networks that are pathological using graph theory. And here we did this using amyloid PET. So we know that amyloid pathology is important for Alzheimer's, and PET is a very good way of introducing the radio tracer that will bind to amyloid. And you can see here on this slide, on these hotspots where the amyloid pathology is located. So we had this for a very large sample of, so they have these images for a very large sample of non-demented individuals. We analyzed the images. We co-registered them to a brain atlas in order to extract from each region the levels of abnormal amyloid for each of them. These regions were then represented as the brain nodes according to graph theory standards, and then we correlated, or we estimated the connections between them using correlation analysis, which is not a real, as Peter said, connection, but it's a way of estimating whether two regions show certain levels of similarity. There are different theories. I could say that I am a proponent of structural covariance. I think it works pretty well, and also for clinical purposes, but there are people that feel differently, and we can talk about that. So one of the things that we found that we thought were quite interesting was that in early stages of the disorder, using this pathological network, we already found a lot of changes highlighted here by all the nodes. And these changes, what they show is that there were more direct connections between all the regions you can see here in early stages of the disease. And if you notice these regions, they lie along the mid-parasagittal line, which are amongst the regions that show more amyloid pathology. So this gives us an insight into how perhaps the pathology is spreading in the brain, no? So pathology might be spreading through hubs or might be spreading through regions that are directly connected. So this is what we concluded from this study. When it comes to the clusters of connections, which I also mentioned before, we found this measure to be very useful to detect different stages of Alzheimer's disease. So here we build gray matter networks using all the regions at the cortical level and the subcortical level that you can see here. And we had 4 different groups that we wanted to compare to controls. So these groups corresponded to different Alzheimer's disease stages. 3 of them had cognitive impairment, but one group was relatively stable. It had cognitive problems, but you could still go out, do the groceries, keep a certain level of independence. The second group was a little bit worse. Because after several years, that group, we found out that it developed dementia. And the third group was the worst of them all because it developed Alzheimer's disease after just 2 years. And for comparison, in the last column over there, we also included a group of Alzheimer's disease dementia cases. So cases already diagnosed, people that are not functionally independent and need a caregiver with them almost all of the time. So when we analyze the normalized clustering between these groups, we could see that it was decreased, as denoted by these orange circles going outside the confidence intervals, which were calculated with permutation testing. They were decreased across the first group, the second group, the third group, and the fourth group. So the clustering seems to be something that changes or is altered across all Alzheimer's disease stages. And the last measure that I wanted to mention is the communities or models, which also seem to be very important to detect the very early signs of Alzheimer's disease. Again, here we had a very large cohort of individuals who, despite being elderly, they were all functioning normally. But in the first group, which was the group without Alzheimer's disease risk, we found 4 clearly different communities. One in orange comprising the posterior brain regions, one in yellow comprising frontal regions, one in blue comprising temporal regions, and one in purple, which seems to be scattered, but it's in fact located across the mid-parasagittal line. So here there were 4 models that were following an anatomical organization. When we look at the group that was cognitively normal but had signs of AD pathology, we found only 2 models that did not seem to follow any kind of brain organization. And actually, when we quantify the modularity, we found that it was really decreased in this group over here. So this is another measure that we could use to potentially identify the very early stages of Alzheimer's. What about other disorders? I don't know if you work with healthy populations, with patients with specific disorders, but this is a natural question, right? So Alzheimer's disease is a very severe disorder, but it's not the only one out there. So it's the most prevalent one. And the second most prevalent one, it happens to be Parkinson's disease. So Parkinson's disease is best known by its motor symptoms. You know, the resting stroma, the patients present, some of them have problems in moving the muscles of the body. Others show some postural instability. All of this because they develop a pathology, alpha-synuclein aggregates that form these well, beautiful, but not that beautiful, of course, Lewy bodies in the brain, which damage a lot of different things. But one thing we know is that Parkinson's disease is no longer just a motor disorder. It's a disorder that in which we can also find cognitive impairment from early disease stages, which increases with disease progression and also increases a lot the risk of developing dementia in the future. So it's quite a relevant group, right? If we want to target it with medication, with clinical trials, this is the group that we should target. And one of the things that we observed was that when we build the networks for this Parkinson group with cognitive impairment and compared it to Parkinson patients that were cognitively normal in a group of controls, we found that the strength of the connections were significantly decreased. And you can see here by the binary graphs, right, how disorganized the connections were compared to the other 2. When we took a look at graph theory measures, again, the distance seemed to be a very good measure to detecting changes in this disorder. So patients with Parkinson's and MCI had a larger distance between their areas. And at the level of single regions, this distance seemed to affect mainly areas from the frontal and the parietal cortex, which, as you know, are regions that are very important for executive functions, such as decision-making and planning, and many other activities that we carry on in our daily lives. We also measure the hubs in the same group of individuals, and this again confirmed that there was a loss of brain hubs in patients with MCI, particularly in regions that we classify as heteromodal, because these are the regions that are involved in complex cognitive functions. In another study, we were also interested in seeing whether the brain models were affected in Parkinson patients with MCI. And although they had the exact same number of models compared to controls, we could see that they were slightly disorganized. So there was the fault network here in blue, which was really, really large. There were some nodes of the cerebellar network that were part of also the default mode network and other regions in between. So there was some reorganization. We could not really evaluate whether this reorganization was good or bad. But when we took a look at the connectivity strength within each model, we found decreases in patients with MCI in frontoparietal regions in the first case, as well as limbic and subcortical areas. So yeah, there's also problems in the brain models in patients with MCI. So to conclude, this was really long. So the distance between brain areas seems to be one of those measures that is affected across both of these neurodegenerative disorders, whereas other measures seem to be more relevant for one or another. But as you know, if you have any experience with graph theory, this usually depends on the imaging modality that they're using, the group of patients, etc. And again, the communities either were lost or disorganized in both of these disorders. So graph theory it seems to be a very good measure to assess how networks become disorganized in different ways in neurodegenerative diseases. So changing a little bit again the subject, another thing that we currently are working on is on synaptic markers, because this is another way of measuring brain connectivity, and it sort of completes the whole picture for us. And in this study, we were particularly interested in 4 proteins. 3 of them are synaptic. So we had neurogranin, which is a postsynaptic protein related to the number of dendrites, that postsynaptic dendrites that you have. We had also 2 synaptic proteins on the synaptic terminal, so presynaptic, GAP25 and SNAP25 and GAP43. And we also assessed as a control or for comparison the NFL or neurofilament light chain, which reflects the integrity of the axons, which is also somewhat related to brain connectivity. So when we assess the levels or the concentrations of these 4 different proteins across earlier and later stages of Alzheimer's disease, where earlier stages comprise individuals with only amyloid pathology and later stages comprise individuals with both amyloid and tau pathology, we observed that all of the synaptic markers were significantly increased, indicating there was more synaptic dysfunction in earlier disease stages, whereas in contrast, the axonal mark NFL was only changed in later disease stages. So we thought this was quite interesting, no? And again, we had measures of functional and anatomical connectivity, which we related to our markers. And we found that synaptic markers were associated with the functional MRI signals in the anterior and posterior parts of the default mode network, whereas the axonal marker, much to our disappointment, was not associated with anatomical connectivity, but instead it correlated with tau PET, which again, correlated with the integrity of the anatomical tracts. So to summarize, in this study, we found that synaptic markers change in earlier stages of the disease, being associated with functional connectivity, whereas the axonal markers change in later stages, being associated with tau pathology, which in turn is associated with loss of anatomical connectivity. So this study comprised amyloid PET, tau PET, diffusion-weighted imaging, fMRI, 4 different proteins extracted from the CSF. And I've not shown all of these results because also the different markers were associated with different cognitive functions. But I think this is what got it the prize for the best neuroimaging article in Alzheimer's disease in 2021, which I'm of course very proud of. Future directions. So I bored you long enough with all the things that we have done in the past, but as you know, this field is constantly evolving and also our research interests. So we must not stay still and also try different directions. So one of the things that we have been doing that I find particularly promising is to understand brain connectivity by building multi-layer networks with my PhD student, Anna, who is sitting here in the audience. And here, a multi-layer network is a network composed of different layers in which each layer, in this case, corresponded to a different imaging modality. It can correspond to other kinds of data, but it's normally used by combining different imaging modalities, and it looks pretty much like that figure that Anna did. So in this study, we took a look at a multi-layer network one that was composed of amyloid PET data and the other one of gray matter data. And when we assessed the overlapping connections between the two layers, we found that there are already changes in the earliest stages of Alzheimer's disease, so cognitively normally subjects with signs of AD pathology in regions that are very important for the early stages of this disorder, because they are actually important for memory functions. And again, when Anna went to the next stage, so people with cognitive impairment and amyloid pathology, she found changes in the same brain areas, which extended to additional temporal and occipital regions. In another study, Anna built a multi-layer network, not composed of different imaging modalities, but composed of different time windows within the whole resting state fMRI scan. So she did this across multiple time windows, and she measured different organizational measures across these time windows, and she did this for a group of controls and patients who were at different stages of Alzheimer's disease. And one of the most relevant findings, in my opinion, Anna might disagree, is the fact that we obtain really good classification performances for distinguishing different disease stages. And as you know, fMRI is an invasive imaging modality, which could be better used in the clinic if it's analyzed properly by taking into account its temporal variability over time. So, for example, for the first group, the group that was less impaired, we obtained a UC of 0.90, for the second group of 0.92, and the final group 0.99, on top of age, sex, and other measures. So we thought these results are very, very promising, and might actually bring to the field to recognize fMRI as a biomarker for many of these neurological disorders, which is not the case for Alzheimer's at the moment. So I do have experience with MEG, not a lot of it, as you will quickly realize by the way I'm going to explain this. But here, thanks to a collaboration with Thomas Junot from Lille University in France, we got some data from MEG and also some diffusion weighted data. And Thomas asked us to analyze this using a multi-layer approach in which we had a structural modality or a structural layer and a functional layer. And he asked us to calculate a measure that, let's say, that calculates how evenly connected the same region is in the two layers, whether a certain brain region is a hub in the two layers, for example. And this analysis showed that this measure was decreased for the inferior temporal gyrus, and it was increased for the supramarginal gyrus, if I remember correctly from my previous piece of knowledge. And both of them, increases and decreases, were associated with better global cognitive function, which basically means that some of these changes might have been compensatory and might allow individuals with older age to still maintain a certain level of cognitive functioning. So another direction that we are currently exploring through my postdoc committee is what we call directed brain connectivity, and it's simply based on the fact that we know from previous literature that brain regions are not all activated at the same time. And typically, some brain regions are activated first and followed by the activation of others. It's a physiological process that is very well established. And in order to capture this process, which is something that is not currently done in fMRI analysis, Mita, what he did is that he assessed the relationship between the activity between different regions after a certain delay, because that delay exists in real data, right? So he defined the delay according to the repetition time and assessed it after 2 seconds, 4 seconds, 6 seconds, and so forward. And he also combined it with the graph view measures, the clustering and the efficiency, which is a measure that is closely associated with the paths. And he was able to detect many differences across different Alzheimer's disease continuum groups across different delays, which also happen to be very closely associated with cognition, such as attention, executive functions, and memory. And by the way, this paper is no longer in review. It has been accepted by Alzheimer's Research and Therapy. Another thing that we are exploring is the application of deep learning to understand better connectivity changes in Alzheimer's disease. So here in this study, a student of ours, UA, developed a new approach for deep neural networks that allows taking into account missing information. So if you're dealing with clinical data, well, some of you at least, you might be aware of the fact that some patients, some individuals do not complete all the tasks, or they might drop out from a study. And so you have to deal with missing data. What most people do is that they exclude the subject with missing data, or they use statistics like imputation to replace the values of the subject by the mean of the whole population, which we know it's sort of an artificial technique that can introduce biases. So here in this study, he built a neural network using 3 imaging modalities. And here within this black rectangle, you can see the information that was present across all the subjects for the 3 imaging modalities. Whereas in green, you can see the follow-up scans for the different individuals, which were quite good in certain cases. But in white, you can see all the missing information. So for example, for MRI, we had very little follow-up information. So by training the network using both the information that coincided for all the subjects and the available information that was present for the others, he was able to discriminate better 2 groups of individuals, controls and Alzheimer's continuum individuals, obtaining an AUC that was superior to a vanilla network, that was superior to networks built on the separate imaging modalities. And here we can see just a heat map showing the most important regions. And by the way, his method was better in terms of performance to a network that used imputation statistics. So if you have missing data, I suggest you take a look at it, because it's a really cool method. So obviously, most of these analyses were conducted using your own software, which is called BRAF, and it stands for Brain Analysis with Graph Theory, not the most imaginative title, but the only one we could come up with. And it's a MATLAB-based software freely available to download at braf.org, while you'll find different information like manuals, videos, and also support forum that allows you to get quick answers to questions you might have within a few days. This is the first version of the software, which was published a couple of years ago, well, a few years ago, and it has been highly cited. And we are now very excited about the second release of the software, which will include the latest developments in the field, such as multi-layer networks and deep learning analysis, which in my opinion are going to revolutionize the way we analyze brain connectivity. BRAF is also on GitHub in case you're interested in expanding its functionalities and becoming part of our community. We're very open to collaborations in case you're interested. And we also posted on Twitter recently just to show that if you're interested in being updated with the latest developments, then you can follow us. And last but not least, I would like to thank the team behind BRAF development, which is a collaboration between Karolinska Institute and Gothenburg University, my lab, and Giovanni's Volpe lab, and also the different sources of funding, and a lot of collaborators that would not fit on this slide, unfortunately, that I couldn't list here. And of course, all of you for listening without standing up for another cup of coffee. That's impressive because I really need some caffeine. So, yeah, thank you.
Thank you, Joana. Are there any questions?
Thank you, Joana. It's a very nice presentation. I've learned a lot. I have actually a question regarding the first part you have showed. That's you have observed a group of individuals without any cognitive problem, and then you measure tau pathology in this group, and you found out, I think the most interesting part is you found out that tau pathology is connected to diffusion tensor imaging markers, like reduced.
No, it's not the same study, unfortunately.
Okay, can you show that slide?
Yeah, so the very first thing I showed was the patterns of, no, maybe you're speaking about this guy. Sorry.
So at very beginning.
Is that the beginning?
Yes, it's before.
Before.
No, it's earlier, like very first.
Earlier?
That first. Yeah, especially go back.
Maybe this slide.
The one have conclusion. That's you show the special correlations between tau and
this one? And resting state
and the DTI markers.
Yeah?
Yeah, that one. So you only found tau but not A beta.
Yes.
So that's actually pretty interesting because, you know, white matter integrator is kind of like also related to vascular pathology.
Absolutely.
So how would you treat the vascular lesion, like white matter lesion in this study?
You know, it's very controversial, the contribution of vascular pathology to Alzheimer's disease, right?
Yes.
This finding, I would simply explain it because tau, in its good days, it's a microtubule associated protein, which serves to maintain the structure and the rigidity, or let's say the morphology of the microtubule. So when it becomes impaired, it means that it's dismantling the microtubule, and microtubule, perhaps I should have been more complete. Microtubules are the structures that are within the axons. And here what we are seeing is measures of white matter connectivity. What are these measures? These are axonal measures.
So there's
That's
no
why.
that. Okay, I see.
That's why. That's why tau predicts very well. And that's why there is a lot of assistance. I also, from my side, I must confess, I recently got accepted a paper that you will see it probably in a couple of weeks, in which we discuss a lot the spread of brain pathology through brain connections. And one of the possibilities would be through anatomical connections, because those are physical elements that are present in the brain. So they're not like functional connectivity, in which you can have activation between three areas that do not have an anatomical connection between them. So we wrote in a recent review that the pathology could potentially be spreading through the axons and passing on to the next neuron through the presynaptic terminals.
Very excited finding.
The prion hypothesis. So we suggest two things: that maybe the connections are passive conduits. They just pass the pathology from one neuron to the other, and from one large brain region to the other, or they may have a more active role. While tau is spreading, it might be that the connections get deteriorated themselves. Or in another case, they could be accelerating the role of spreading of pathology. So we discussed these different views. And if you're interested, I welcome you to read it.
Definitely, thank you.
Not because it's my paper. But,
may I allow to have a second question?
of course.
So you talked about the graph theory things. I'm just wondering how graph theory could handle like repeated measurements for different time points. Like if you have
Very well, actually.
very well. So how I'm thinking about is that correlate, because I don't know this method. So is that you're seeing the correlations from each time point, or you're making the change?
So if you have longitudinal information, what you can do in the most simple way is to build a brain network for each time point and see how it changes over time. And then the permutation testing would not be done within two groups, between two groups, but within two time points, which would make it a little bit easier to get the statistically significant values, like other softwares have done.
That's very interesting.
Yes. Yeah, no problem.
Questions? Online questions?
Yeah, about this imaging, I was curious, like when you, because you inject in the blood,
Yes. We make patients suffer.
so I was thinking, because is it clear that blood barrier is a bit disturbed in Alzheimer? Because
It is.
so I was thinking, like, is it possible that you see more of these synaptic ligands because of this, or is it something usually?
So there are a few biomarkers that you can extract from the cerebrospinal fluid, such as albumin, that reflects the integrity of the blood-brain barrier. There's also imaging measures that you can use nowadays with arterial spin labeling that can show you how, not affected or preserved the blood-brain barrier is. It is possible that the tracer passes more, but it's not something we are worried with, because it seems to pass in all cases, and in all cases it seems to bind. So if we had seen a difference in the binding, we would probably had related it to the blood-brain barrier, but we don't see it. And the blood-brain barrier is important because, of course, if you don't have that protection barrier, many bad things can pass to the brain. For example, viruses, which have been shown to also to increase a lot of pathologies in the brain and increase your risk of dementia. But for the blood-brain barrier, specifically in relation to PET imaging, I've not seen evidence that it's so the tracers that were developed specifically to cross the blood-brain barrier, both in healthy individuals and in individuals with Alzheimer's, is producing different results. That's my opinion, at least. I think we're all tired. No?
I can ask a second one.
Of course.
So I just maybe if you can discuss a bit more on how you interpret the results of the changes in network with your. Did you do the link between change in network and the change in this synaptic?
Oh, the study, my favorite study, the synaptic changes.
I mean, do you?
Maybe I got overexcited. You meant something else, right?
No, I think the synaptic one, yeah. But did you link it to
Oh.
the network changes? That was my.
What is your question?
Did you link it to the network changes that you observed? Like,
Yes, we linked it to many different things. So our main aim in this study was to try to see what is happening in earlier disease stages and late disease stages. Because we know that earlier disease stages are associated with amyloid accumulation and late disease stages are associated with tau accumulation. But there is not much evidence in human individuals that explains why there is a delay between these two events. There is a temporal delay. And how are they linked? What is happening in the middle that drives one thing to the other, right? So in this study, we tried to address this using many different measures. And what we saw was that all our results. So I didn't show you the best picture of this paper, which you all done, I mean, which shows that after amyloid starts accumulating, what happens is that the number of postsynaptic spines starts decreasing, and the number of vesicles in the presynaptic terminal starts decreasing as well. And after that, we have the increase of tau pathology, which starts disintegrating the microtubules because the tau becomes hyperphosphorylated. and that means that it's not anymore is no longer able to keep the microtubules where they are, and this leads to the degeneration of the axon. And after that, you develop cognitive decline. So one of the things I did not mention, because I didn't want to confuse more the picture here, was that also these changes, so early disease stages related to amyloid were associated with functional connectivity, were associated with synaptic markers, which were associated with functional connectivity and were associated with memory functions, which are the first symptoms. Whereas late disease stages associated with tau pathology, the axonal markers were changed. Also, global cognition was changed. But when it comes to anatomical connectivity, the main driver was tau and not the axonal marker. And that's because of the size of the structures they are reflecting. Because neurofilament is reflecting the very tiny filaments within an axon of a neuron, whereas tau pathology is actually reflecting the microtubules, which are, I don't know how many orders larger than these tiny filaments. So yeah. It was a fun article to write, because everything seemed to fit, which is not